more about selective inference?
So I would like to begin by thanking the organizers for sending me an invite to this workshop. I'm very happy to be here. And I'm here to give you a roundup on some recent activities in selective inference, which are centered around construction of interval and point estimates. So this is part 1 of the tutorial. And my goal here is threefold in nature. First, I want to give you examples which will probably and hopefully motivate you as a data curator to understand that some of these tools of selective inference that I talk about today can be deployed if you really ever want to combine some of your exploratory findings with a confirmatory analysis. Second, I want to start talking about a cutting-edge application in genomics where I deployed selective inferential tools to get effect size estimates post-data mining. So my goal here is not really to tell you about the application that I worked on, but rather to tell you that there can be many more useful such applications where we can use conditional inference to enhance our understanding in those communities. And thirdly, I would start by talking about some first approaches which are aligned along a conditional perspective to selective inference. Hopefully tell you what are their shortcomings and how we have overcome them and where are we right now currently. So to start with, we all know that naive inference or classical inferential regime starts with the basic assumption that the analyst always knew the small set of variables that mattered from thousands of noisy variables. But we have that understanding so far that this is far from reality. And in reality, what we do is once we have the data, the analyst explores the data to find some meaningful structure in it. And that's when, if he wants to calibrate the effect sizes of those discovered findings, he would need some sort of selective inferential tools to come to his rescue, because no longer classical inference will give you the guarantees that you want to have. So selective inference is a broad field that tries to blend in confirmatory exploratory analysis with the confirmatory paradigm. And like I said, it's a pretty broad term, and there are different lines of work that aim at different sorts of guarantees depending on what your goal is at the end of the day. So let me start with a toy example. So you have a response y to begin with, which is n-dimensional, n being the number of samples you have. You have some predictors, a set of p predictors to be precise, measured for your n samples, recorded in your predictor matrix X. You run, say, our favorite lasso query coming from the Sequoia Hall to find some set of predictors which hopefully explain the variance in this response y. And you now want to report inference for the active coefficients that Lasso gave you as an output. So let's see a concrete example. So I'm giving you the data generative mechanism. Say, for instance, here n is 500, so I have 500 samples. My p is 100. I generate x as a matrix with Gaussian entries with covariance given by this covariance matrix that has correlation parameter 0.2. I generate my response as just noise, which is completely independent of the predictor matrix. It has nothing to do with matrix X. I just run Lasso at a fixed value of lambda. Typically, I take this lambda to be a theoretical value of lambda that gets you consistent selection. So this is the active set of coefficients that I see from Lasso, and Lasso shrinks some other coefficients to 0. Now say I want to get inference. One way is that now once Lasso has given me this active set of coefficients, I'll refit a linear model to the selected subspace of X and my response Y. So I run an OLS based on X, E, and Y. So what would be my naive point estimator? So my naive point estimator is the OLS estimate, which is just this. And what are naive confidence intervals here? So naive confidence intervals could be just centered around the OLS estimator and scaled by the variance. So if I report a confidence interval for the jth coefficient of this of a selected target, then it's centered around the corresponding jth coordinate of the OLS estimator, and the variance is just a scale of xe prime xe inverse. The diagonal jth diagonal entry of this matrix is just OLS like regular point estimation in classical inference, where we know that these naive intervals happen to be Gaussian. Now, say you repeat this experiment 50 different times, and you construct your intervals based on this algorithm that I told you right now. And you assess the coverage of your intervals for these reported targets that I told you. And you'll see that the naive coverage is just 30%. So this is not a phenomenon which is restricted to the setting where there is all noise. But if we even put some signals, say now I simulate my y conditional on x with some signals. I have 5 true signals, and I vary the SNR, and I look at the coverage of these intervals. I repeat the experiment in the similar fashion. It's just my data generative mechanism is now different in the sense that I have some signals in my model. Still, the coverage is pretty bad. So naive, sorry?
SNR is variance explained, or?
SNR is the signal to noise ratio, and it is computed as basically if beta is your vector of is your true vector of signals, then it's beta prime beta by this noise. Noise is here 1 over sigma, yes. And this is related to proportion of variance explained as, I think SNR by 1 plus SNR is proportion of variance explained. So this is very low proportions of variance explained. 0.5 corresponds to around 0.5 by 1 plus 0.5, which is 1/3 of the variance explained. So my goal here is to talk about getting valid confidence intervals, and hopefully I'll move to point estimation in the second part of the tutorial. So the first question here is that, okay, our classical guarantees of coverage do fail us. So how do we redefine guarantees for coverage post-selection? And there have been 2 directions of work that have mainly focused on providing valid confidence intervals post-selection. One is the simultaneous guarantee dating back to the Posey paper in 2013, which gives you a control on the proportion of reported intervals that don't cover the true underlying parameter. And that's control at some alpha. But the challenge here is, of course, in determining these confidence intervals. How do you construct these confidence intervals? And the way they construct the confidence intervals, it's in a very general fashion, in the sense that these confidence intervals actually hold for any selection procedure. So if you don't have to tell the statistician what selection you really did on your data to get these confidence intervals. But as you can imagine, that this generality also comes at a huge cost of power. So these intervals happen to be very long. And the second issue is tractability. So I don't think there has been much progress since this paper in making this method really useful so that people can use it. So these confidence intervals cannot be constructed for really like even regression dimensions greater than 30, if I remember. I don't know if they've made some progress in increasing it further, but like I know, at least for real problems, you cannot construct these confidence intervals. So the second perspective that I'm going to focus on is the conditional perspective to selective inference, which has been, I think, introduced more in these papers, Lee et al. And Fidian et al., where the guarantee you look at is the proportion of intervals that do not cover the true underlying parameter conditioned on the selection event. So you try to control that rate. I'll talk about this guarantee in slightly more details a slide later. But as you can see, like to construct these confidence intervals, you need to tell the statistician what selection you applied to your data. So this perspective is quite tailored for the selection process that resulted in you observing this active set of coefficients. But at the same time, it's more powerful and also tractable, more tractable than the Posey paper of Burke et al. And I'll tell you more about how we have used some of these procedures to a real-life application where we have a regression dimensions in the order of millions. So as you can see, the main difference, one is, of course, the simultaneous guarantee of the Posey paper is more general. You construct the confidence intervals without even knowing what selection you applied to the data. Whereas in the conditional perspective, you really need to prescribe what selection you did on your data. But I don't view it in a negative sense, because I feel like if you really want inference with some reasonable power at the end of the day, the least I deserve to know is what you did with the data. So you have to understand what tools you use to explore your data, and you just have to tell me what you did.
Telling you what I did sounds simple, but
it might not be at times.
I could give you an encryption of what I did. What are you going to do with that? So what does it mean to tell you what I did, and how is that specified?
Yeah, so here I think so far still we need to know explicitly the selection procedure that you did on your data. Now, when you say implicit, you also have to tell me what you really mean by implicit. Like, are you still concealing me from me some part of the selection that you did? But so far, I think whatever we are doing is based on the exact selection that you applied to the data. So you have to tell me what all you did to explore across your data.
I don't want to belabor this now, but are you saying that if I used a program to do my selection, I can hand you that program? No,
I mean, you just have to maybe describe to me what that program is. Like you have to, like in the case of lasso, you just describe to me, okay, you ran a lasso algorithm. So you have to tell me what algorithm you ran.
I minimized something. So that has, in some sense, a closed form that's independent of how I actually do it. Okay, never mind.
In principle, yes. It might be hard to carry out. It might be computationally bad.
I don't even know if it's well defined. That's what's bothering me.
We just need to, so we have to be able to describe the event on the event that it's a subset of the sample space that corresponds to making that same selection.
That helps. Thank you.
Yeah.
So the other selective inferential guarantee that we have heard in this workshop and all of you are aware about is false discovery rate. But I want to tell you that it's a very different selective inferential guarantee to me. There is a long line of work that has produced different methods that try and control the false discovery rate. But for me, FDR is really a guarantee on selection. And like Chiara put it yesterday in her talk, it's more sort of a principal way of doing your exploratory analysis where you get findings which have some guarantees, but you still want to investigate them further. It's not a confirmatory analysis exactly. And I'm going to talk about the second part more. So even if you have a procedure that really guarantees you FDR on selection, you ultimately, at the end of the day, have some findings that you want to investigate further. You cannot, like if you still try to construct confidence intervals using classical methods, you do not get the coverage guarantee. So FDR guarantee doesn't really give you coverage guarantees if you are going with a confirmatory analysis. Now let's look at the conditional coverage guarantee a little more closely. So this was the guarantee that I showed you on the slide before. So what does this mean explicitly? So if I try and construct 90% confidence intervals in the conditional regime, then I expect at max 10% of them to not cover the true underlying parameter when conditioned upon the selection event. And this is what is dubbed as selective false coverage rate control in Fiddian et al. So this guarantee is what we call as selective FCR, and selective FCR also implies control of PFCR, which in turn implies control of FCR. Now let me just define what FCR and PFCR are, just in case people are not aware of them. So FCR has been defined in this paper by Benjamini and Yacutelli. It's the proportion of intervals that do not cover the true underlying parameter amongst the reported intervals. So you report a certain number of intervals after your selection event. It's the proportion of intervals that don't cover the true underlying parameter. And PFCR is nothing but FCR, because FCR, this quantity might not make sense when you report nothing, when mod E is actually 0. So PFCR is just FCR conditioned on the fact that you report at least something, that mod E is greater than 0. And we can clearly see very trivially that FCR is actually PFCR times the probability that you report something. Now, just maybe a little slide to show you that selective FCR does imply control of PFCR. So it's a more stringent control. So if you do control selective FCR, you end up controlling PFCR and you end up controlling FCR as well. So we start with the definition of PFCR. Like I said, it's the proportion of intervals, reported intervals that don't cover the true underlying parameter conditioned on the fact that you report something. If we use the Tower property of expectation and condition it on the selection event, then what this turns out to be the sum of the probabilities that the jth coordinate of, or the jth confidence interval doesn't cover the true underlying parameter conditioned upon selection. And this is what this conditional inference tries to control. So it controls this quantity at level alpha. So if you do control this quantity at level alpha, you can see that what you have is the sum turns out to be alpha times the number of reported intervals, and you end up controlling PFCR. So it's not very hard to see that selective FCR actually guarantees you a control of FCR. So now that we have a sense of what the conditional line of work is really trying to control, let me digress a bit and give you an application in genomics and tell you why we need such tools to improve our understanding of effect size estimation in such applications. So the application that I'm going to talk about is related to expression quantitative trait loci studies. These are called EQTLs in genomics. They try to identify regions of the genome that tell you whether or not and how much a gene is expressed. In particular, I'll be looking at a cis-EQTL mapping. So a cis-EQTL mapping in particular tries to find associations between gene expression, which is the response variable. So in terms of statistics, if you want to know what the response variable is and what your predictors are, well, the response variable is your gene expression. And the predictors are some local genetic variants located around the gene. So what do I mean by local genetic variants? So you basically measure SNPs. You take a gene, you look at SNPs within 1 MB of the transcription start site of the gene. So you look at a local window around the gene, and those constitute your predictors. So this is maybe a small example to illustrate it. So here is a gene whose gene expression you have recorded. That's your Y, or your response. And this is the small window that you move around the gene and record all the SNPs around this gene. And these are your predictors for the gene expression of this particular gene. So to give you an idea of the data set that puts this whole application in context, so this is GTEC, it's called the GTEC data set. It's associated with a GTech project, which was an NIH-funded project, and you can find more details about this data set here. There are many more details about the data set, their findings at their portal. So briefly, what they look at is 44 human tissues, and there are 449 donors in all. So these are human tissues. You have collected gene expression data for genes corresponding to 44 human tissues, and the predictors are recorded from the genotypic information that was collected from the donors. But if you look at specific human tissues like, say, liver or brain or stomach, etc., you will notice that the number of samples is actually even smaller than the number 449. That's because each individual actually contributes to samples for only a subset of these 44 human tissues and not all of the tissues. Further, you can see that a big challenge in this EQTL, human EQTL study is the limited samples associated with each tissue. That is because no healthy individual is going to give you a part of his brain to advance science. So you see just 72 samples corresponding to brain, and that's a huge limitation. So from now on, maybe let me just focus on one tissue, which is liver, which has 97 samples. So to cast the data into a statistical framework, we have roughly 22K genes in the genome corresponding to every tissue, but we are just focusing on liver for now. So we have 22K genes. Each gene, so this is the gene expression, say we focus on gene G. So this gene G has gene expression recorded as Y, and it has a predictor matrix XG, which consists of all the local variants around that gene. So we have this Y matrix, which is a 97-dimensional vector, 97 being the sample size, and this Y matrix comes with its own matrix of predictors, which are the local variants around the gene, with number of columns being the number of variants around that gene. So this number typically varies from 3,000 to 25,000. So that's your P. So you can see that this can be summarized as a multi-phenotype analysis, where you have multiple phenotypes, the phenotypes being the gene expressions associated with the 22K genes. So you have 22K many regression problems, where each regression problem comes with its own response, Y, and a predictor matrix X. And the size of the problem, like I said, N is 97 here, and P varies from gene to gene, a number between 3,000 to 25,000. So to give you a brief idea or a bird's eye view of how the selection of regulatory variants proceeds in these studies, like what is the state of art of selection, it's actually modeled as a two-stage process. So you want to find regulatory variants that regulate the expressions of these genes. So first of all, from 22K genes, you try to select genes that have at least one signal around them. So each gene has a set of local variants around it. You try to identify the genes that have some signal around it in the first stage. So these genes are called E genes. So from 22K genes, you try to select the red ones. So you select a subset of these 22K genes, which have some promise of signal. And in the second stage, once you have scanned out the genes, E genes, you look a bit deeply into the local variants of these selected E genes. And you run a multivariate regression problem for each of these E genes, and report variants corresponding to each of these selected E genes. So
when you're counting down on the bottom row, V1, V2, this would be up to VP?
Yes, up to VP, exactly. So these are your variants, and you're trying to focus your computational resources into looking deeply around these selected E genes and report the regulatory variants that are associated with each of these E genes. So these variants that you report in the second stage for each E gene, they're called E variants. So this is just a bird's eye view of how the selection is done, and this is a well-documented process already. It comes from the GTech community, and they have spent a lot of efforts in trying to document how you mine this data to get to E variants or regulatory variants that tell you that hopefully are findings that regulate gene expressions. So once you have found these E-variants, the next logical step is to try and estimate the effect sizes of these E-variants. But of course, the catch here is that you could have encountered potential selection bias in having mined only about 5,000 E-variants from roughly 1 million variants to begin with. So there could be heavy selection bias when you try to calibrate the effect sizes of your discovered associations. So the question that we can hopefully try and answer in this application is that how do we estimate the effect sizes of potential E-variants that have been discovered through a genome-wide search over the space of predictors?
Sorry, one more question?
Yes.
So P wasn't anything like a million. Where's the million coming from?
Oh, it's a multiple, summing over all the regression problems, yes. Now to just show you, I'm sure you're convinced, but to show you how naive inference can lead to completely misleading results. So this is a simulation that I conducted on the scale of GTEC. I used the genotypic information from the donors from the actual GTEC data, but I simulated the responses on the scale of this data set. And I followed the two-stage procedure documented in their paper. And I just did naive inference trying to construct confidence intervals and report their coverages. So the x-axis here is denoting the different signal regimes. So 0, for instance, is the regime where genes have no true signals around them. 1 is the regime where genes have exactly one signal around them, and so on. And these are the coverages of the intervals reported for these genes. And you can see that naive intervals are all short of the aimed coverage, which is 90% in this case. And they fall short by a wide margin. So we really need to do something about such applications for sure. So
quick questions.
Yes.
So in this example, you actually have 3 stages, right? You first select for genes, and then
2.
those genes, and then the variants.
Yes, that's it.
And the variants you compute. Okay, so you consider those to be the 2 stages.
They are the 2 selection stages, yes. It's a hierarchical selection. You select.
And in the first stage, to identify the E genes, are you testing for all of the variants?
So I'm going to give you more details about that screening state. So I did not tell you how E genes are really selected. So for each gene, what they do is they compute the marginal associations with all the local variants around the gene. It's a marginal screening process. And then they do a global null hypothesis that there is no signal around it. So you can do something like the Symes test or the Bonferroni to test that that particular gene has no association with any of the local variants around it. So that's based on the marginal associations of that gene with each of the local variants. But you are not taking into account the correlations coming from the other variants around that gene. So it's typically done as a marginal screening procedure because they don't want to invest too much computational resources at that stage. And then once you have narrowed down your genes to your set of E genes, that's where they started doing a second stage of screening where they ran a multivariate regression for each of the E genes, trying to identify those few regulatory variants around each E gene. So now, after having talked about this application a bit, which I'll come back in tutorial 2, I want to start by discussing some first approaches to overcoming selection bias, which are aligned along the conditional perspective to selective inference. So I'm sure this is something that all of you must have done at some stage of your confirmatory analysis, which is data splitting. You know that data splitting basically overcomes selection bias. You explore your data on a random split of the data, but then decide to discard it completely and use the remaining fraction of your data to report inference. The question is, does data splitting control selective FCR? Well, it does. If you were not aware of it, it does control selective FCR in the sense that if you assume that the data points that you conducted your selection on is independent from the data points that you're using in your confirmatory analysis, then this conditional probability is just equal to the unconditional probability, and you do control it at alpha. So it's certainly a very appealingly simple procedure that does guarantee you selective FCR control. Now the question is, we are completely discarding the data that was used in selection while we do data splitting. So do we have room to still use some leftover information from data in the selection stage? And the other question is also, can we always afford to conduct data splitting? Like in the EQTL example that I just described to you, where you barely see samples in hundreds, is data splitting even a reasonable option that you have? Well, no, because if you did do data splitting, conducting selection on, say, a random 50% split of your data, you end up discovering only 4% of the e-variance that the baseline GTEC paper reports. So you don't even find enough interesting effects, forget about inference. So your only bet would be to maybe wait for new data points to come up. And this project, if I'm not wrong, has been going on for 5 years now, and there is no new data set. So, well, if you really want to calibrate the strengths of your discovered associations, is the best bet that you have is to wait until eternity till you have this new data set. So we really need to think of ways of trying to do something more reasonable than data splitting, where we still discover the same findings, yet have some reasonable power when we report inference. Yes?
I have a so, sorry, what are you comparing to here? What's the 4% of?
Oh, so this.
Kind of something that's like not statistically.
So I was analyzing the GTEC data set. So it's a consortium that is mainly aimed at trying to find these regulatory variants, which you want to investigate further. So they don't calibrate the effect sizes of these variants, but they do try to do hypothesis testing and find these regulatory variants to begin with.
But
so
is it it's consistent with everything you've told us that like 96% of what they reported is just, you know, artifacts of selection?
So they don't, actually to their defense, they do not do confidence intervals at all. They just tell you like these are some associations that they discovered from the data, that's all. So they are not doing effect size estimates at all. I said, okay, if on top of their findings, if you were to apply some naive inferential tools to get naive confidence intervals, they are not valid anymore. So to their defense, they don't report confidence intervals to begin with. They just report findings.
I guess I'm asking a different question. Like,
so
like
they used...
Maybe this is a defense, maybe this number at the bottom of your slide is a defense of data splitting.
Sorry?
Like, is it obvious that it's...
Like, is 4% a bad number here? Because
yes,
like,
yes, because...
What I'm saying is, like, it's entirely consistent with the story. Like, maybe everything they're reporting is noise.
No, that's not true. That's not true.
The larger question I'm asking is, you're comparing something that you know is statistically valid to something that you're saying has no kind of solid justification behind it.
No, I mean,
Maybe I misunderstood.
no, let me make it clear. So already I think they say that they discover much less than expected. That's because of those limited sample sizes. So sample sizes are barely in hundreds. So the conjecture here is that they're discovering less and they're limited in their selection power already because of those low sample sizes. I'm saying if you further reduce that sample size by a half, then you are even losing almost all the signals that they discovered. Or maybe, I don't know whether they're signals or not, but you're losing a lot of what they were already reporting. So my guess is you're already losing a lot of interesting stuff to begin with. So when you come to inference, it doesn't make sense because you're not discovering anything interesting as such. So forget about the goal of inference in this case, if you're reducing your sample size to even a half.
Okay.
Like, not to belabor the point, but like a priori, just to understand, like a priori, like suppose I could oracularly discover exactly the sort of correct genes.
Yes.
Would it be obvious, or should it be obvious that that number is, you know,
on the order of the number of discoveries in the baseline, or could that be much smaller or much larger?
It's much larger. So all I'm saying is that you're losing a lot, both in select. Because if I were to do like, so I'll be reporting inference for only these 4% of E-variance, right? And I mean, the short answer to your question is that I'm losing a lot of signals. So I'm not discovering enough of signals, and I'm reporting stuff for much lesser stuff, and I'm losing both power for selection as well as inference by doing data splitting.
Snigdha, just aligned with those questions, so in this regime of work, is there any possibility or any kind of like wet lab tests on other species which have confirmed any E-variance? Like, then you would have some idea of the number of E-variance, like number of discoveries you were expecting. So is there any body?
So this is human EQTL, and I guess this data set, as you can see, is already like, I don't know if there is a second data set on human EQTL. So it's always very hard to obtain samples for humans, yes. That's, so even for this project, I'm sure, I mean, they should try to get newer samples, but they haven't, so there must be a reason for it.
So GTEx is collecting more samples.
More samples.
And they're also doing these CRISPR screens, which are just going to the cells and then modifying the locations with genome editing to test these E-variants.
Uh-huh. So yeah, I don't know of a second study which really provides you some sort of confirmation where you can compare the findings across GTEC and some other study. I don't know of a second study on human EQTLs.
Just to push on this like a little bit more.
Sure.
So when you double your sample size, say, which is what you're trying, effectively want to,
yes.
You're
Yes.
a factor of 2 in the sample size. At a given level of confidence, just sort of naive, I don't know, straightforward asymptotics suggest that like the effect sizes you can detect will go well, down or up depending on which direction you're going by like what, square root of 2 or something like that?
The lengths of the confidence intervals, you mean?
Yeah, I guess that's right. I mean, I'm thinking like
Yeah, sure.
rule of thumb, about over square root of n, so.
Yes. That's, yes, sure.
So I guess I'm, like, given that, is it the case that you think that, like, a ton of these effects are, like, right above that threshold where they're going to be detectable with with a sample size of n, but not with a sample size of n over 2? Because, like, you're fighting for a factor of 2 in the sample size, which makes sense when you only have a small number of samples.
That's true, but you have to,
Is it plausible that you just, like, no matter what you do, you don't have enough data to do the things you want to do, period?
Yes, I mean, you have you have a buffer on, like, how much power you have with these many data points. That's true. But like I said, like, my point here was that I'm not even talking about inference. I'm just saying that you're losing findings to a great extent. You're just discovering 4% of what GTEC discovers with double the sample size.
But like, again,
reports as interesting associations.
GTEC's an FDR controlling method?
There is no theoretical guarantee on FDR control, ultimately on the E-variance that they report. They try to control FDR at the E gene level, for sure, because once they do those global null tests, I'll describe that in details later. They pass all those global p-values through a BH filter. So they do have FDR control at the E gene level, but they do not necessarily have FDR control at the E-variant level, but they try to somewhat empirically have reasonable control of FDR at the second stage, because they try to penalize the fact that you have selected those E genes to begin with, for which you report the E-variance. But they don't have any theoretical guarantee on FDR as far as I know.
Okay, so this is reaching, but maybe one possibly plausible explanation is like pH and other FDR type controlling procedures, and if GTEC is using other things, the sort of macro signal is coming in to the process to sort of help to reject things. So even if there are like still a decent number of small p-values, this is just a possible answer to Aaron's question, even if there's still a decent number of small p-values, there just might not be enough. There's some kind of like teamwork that goes on in pH and other FDR controlling procedures. So it's some kind of...
their selection...
threshold type thing. I don't know.
Like their selection to me, honestly, is still very conservative. I did not try to modify the selection procedure because that was not my job. I wanted to take like the state-of-the-art mining procedure that they do and try and provide confidence intervals and point estimates to calibrate the effect sizes of their discovered findings. So I didn't want to change their selection procedure per se, but if you ask me, I feel it's a very conservative procedure. First of all, they pass it through a BH threshold, which often can be quite conservative. And even in the second stage, they pay a very heavy penalty for the selection that they did at the E-gene level, so they end up discovering a lot less. So whatever is their state of art, I feel it's a pretty conservative selection to begin with. Maybe I should tell you also at this point, like the belief in the biological community is that every gene is an E-gene. So it's sort of bogus that you kind of, you're trying to reserve computational resources by screening E-genes, but the belief there is really every gene does have some signal around it. It's just that they don't have the power to discover it. And also in this two-stage thing, they're trying to basically, reserve computational sources so that they don't have to do the search of regulatory variants at a large scale to begin with. They try to look in deeply for a smaller subset of genes. So now, so this was the discussion on data splitting, but let's go to conditional inference as was introduced in Lee et al. So let's again look at the toy example that we were looking at. So we have a response Y and we have predictor matrix X. As selection, say you just ran lasso to get an active set of coefficients E. Okay, let me just move this way. So coming to inference, you want to, at this point, specify a model and a target that you want to infer about. These are the two objects that statistical inference requires you to specify. So in Lee et al., they assume a saturated model, which is basically assuming that Y is modeled as Gaussian with mean mu. And the target they look at is basically an adaptively determined target that is obtained by refitting a linear model to your selected subspace of X's. Now, for simplicity's sake, say I just look at the first coordinate of this adaptive target, which is the projection of mean mu onto the selected subspace of X. Then if you were to construct naive intervals, you would look at the OLS estimator obtained by refitting your data to the selected subspace of X. And you would, this is assumed to be if you didn't have this adaptive target, you had no selection, then this would be simply Gaussian in this setup. And you have naive inference, which is valid. But since now you're inferring about this adaptive target, the question that we ask ourselves is that how do we construct valid intervals for the adaptive target? Another way of reframing the question is, what is really the right law of this OLS estimator? Since it's not Gaussian, how can we correctly calibrate its law? And this answer was provided by Lee et al. It's based on two key observations. One, the selection by lasso is equivalent to a set of polyhedral constraints. So you can describe the selection by lasso as a set of affine constraints on your response Y. And the second key observation was looking at the law of your naive statistic, which was this thing, which without any selection was just Gaussian. So you try to look at the law of your naive statistic condition on selection, which is this set of polyhedral constraints. And in addition to that, you also condition on nuisance statistics, which correspond to nuisance parameters. So you condition on this p eta per y, which is basically conditioning on all directions orthogonal to your direction of interest eta. So once you look at this selection event, this law happens to be a univariate truncated Gaussian law. And this is based on the observation that if you write out the selection event of lasso, which is the set of polyhedral constraints, and you condition additionally on the nuisance statistic, then your direction of interest, eta prime y, is actually restricted between array. So you can see that eta prime y is just restricted between gamma minus and gamma plus. And these are functions of the statistics that you conditioned on, or basically your nuisance statistics that correspond to nuisance parameters. So you have your polyhedral lemma, as a result of which the law that you are looking at happens to be a univariate truncated Gaussian law. And these gamma minus and gamma plus can be computed explicitly. So this is what is dubbed as polyhedral lemma in Lee et al. Now let's just revisit the toy example of lasso that we have been looking at. It's the same example. So now I report coordinate-wise inference. So I look at this target and I report inference for the first coordinate of it, the second coordinate, third coordinate, and so on. And like I said, the pivot that I use is this truncated Gaussian pivot from Lee et al. Let's see if the intervals now that we construct based on Lee et al., if they're valid or not. So this is the table that I had. So we can see that if we target at 90% coverage, the coverage of the Lee et al. Intervals based on this truncated Gaussian pivot is pretty good. It's close to the 90% target. So clearly it gives us a neat pivot, which is very easy to compute. It's just a truncated Gaussian law. You can compute it. And it gives us coverage guarantees. So the question now is, do we really need to look beyond Lee et al.? Yes?
Let me just understand the form of the coverage guarantees. Like,
yes, it's...
How come the numbers aren't like 90% or above, if that's the...
Why should it be? I mean, the target is 90%. So this is, I'm empirically computing the coverage, right? I'm repeating the experiment 50 times and I'm computing the coverage. So it's close enough to the 90, like anything 2 or 3% plus or minus is good enough. It's just empirical coverage.
That was actually going to be my question. What's the sort of confidence interval width on those coverage numbers? Like,
what am I covering, or?
No, I mean, you're saying each of these coverage numbers.
Yes, so these are confidence.
Based on random sampling of some underlying proportion, which we hope is 0.9.
Yes.
But they're all like consistently right around 87%.
I mean, 0.9 is the asymptotic value. So it's sort of like, 0.9 will hold as p and n go to infinity.
But
No, these are exact intervals. Like you're repeatedly. So these coverages are for the adaptive target that I just showed you. And these are coverages on an average reported over all the selected intervals that you report. And this is something that I compute empirically. So I construct these confidence intervals, see the proportion of them not covering the true underlying parameter or covering the true underlying parameter, as in this case. And it's close to 90%. That's what you expect if the pivot is valid. Yes?
Maybe the question is that...
It's a finite coverage guarantee. There is no asymptotics here. Lee et al. Is a finite coverage guarantee.
So if Lee et al. Is...
It's exactly Gaussian, like I'm generating my data from a Gaussian model as I described to you.
Right.
In such a case, Lee et al. Is exact, and it provides you finite coverage guarantees. There is no asymptotics here.
It seems, I think Adam's question is, if it's actually supposed to be providing 90% coverage, it seems a bit weird that they're all providing 87% coverage.
Like, is there a theorem saying they're supposed to provide like 90% coverage? Because this seems to contradict such a theorem.
Well, except it's based on, you said 50.
Sample, yeah, 50 replicates. Like maybe sorry? 50, yes.
Right, so I guess that was
50 times the number of selected intervals, right? So I'm actually reporting 50 times mod E intervals. So if in the first round I select 5, then I'm reporting 5 intervals. So it's 50 times the average, like the number of selected intervals that I...
Are the Monte Carlo estimates correlated with each other potentially? Like, are you?
This is not Monte Carlo.
Wait. Aren't they? They're Monte Carlo estimates of the true 90%, right? Because you only did a few.
Oh, yes.
Maybe if you use the same noise for 0, 0.1, 0.2, and 0.5, you can get correlated. The puzzle is why are they so close to each other, but not also to 90%? Right?
So let me tell you, I use the selective inference package in R, so feel free to contact the users. I don't endorse this method really, and that's what's coming in part 2 of the tutorial, why I hate this and why there is much more beyond this. So that was coming in the second slide, so I neither endorse the package, it's not something that I coded. So feel free to contact Rob Tibshirani, Ryan, and I don't know who else, Josh, Jonathan. Jonathan also doesn't endorse this, by the way. So, but anyways, I mean, I think they're fairly close enough. So statisticians were happy if they're like, 3% off. Like, I mean, maybe if I do 50 more runs, maybe these numbers look a bit up, down. I don't know, like. Some of them might be close to 90%, so I don't know if this is really a concern for me that they're...
The weird thing is just that they seem like pretty tightly concentrated around 87.
87? Well, they do, but I don't have an explanation for it. I feel the intervals are exact and they should give me good enough coverage.
So there's no approximation?
No, there is no approximation here. It's an exact pivot, and my data generative mechanism is also a Gaussian data generative mechanism, so it should be exact here because okay, so the question that I said is that do we need to look beyond Lee et al? So I showed you coverages of these intervals, but a very annoying part of Lee et al. Intervals is that they can be very long and associated with really low statistical power. So now I'm going to show you the average lengths of these intervals. And if you actually call the selective inference package in R and look at the proportion of intervals that are also infinitely long. So this row tells you the proportion of intervals that happen to be infinitely long, and this gives you the average lengths of the intervals that are not infinite. So you can see that they are terribly long intervals. They don't seem very stable in that sense. And there is a new paper in 2018 by Lieb where he has shown that the expected length of the Lee et al. Intervals are actually is actually infinity. So, so like I said, the takeaway here is that Lee et al. Intervals can be very long. Yes?
So I agree the expectation is infinity, but how would you get an actual infinity? How would you get an actual infinity?
So I just ran the selective inference package, and this is the proportion of intervals that happen to be infinitely long. That's all I'm saying. In my experiment, I gave you the actual data generative mechanism, repeated it, reported intervals. These are the average lengths of the intervals which are not infinitely long, and these many intervals happen to be infinitely long.
Yeah, I just think mathematically I don't understand how that can happen. But maybe it's something about the implementation. I don't know.
So you think it's about the code, but it could be possible, like, that it just happens to be a rare selection event, and that's what we see in Lee et al. Like when you're approaching the selection boundary, your intervals just happen to be very unstable and infinitely long.
This big, but finite, I would
So let's maybe see an explanation of, okay, so this is not a slide on explaining why Lee et al. Intervals can be infinitely long, but this is data splitting for the same experiment. And I mean, I have 500 data points, so I ran the lasso on 250 data points and reported intervals for the remaining 250 data points. And we see that data splitting has also pretty good coverage as expected because it overcomes selection bias. And the lengths are much shorter than Lee et al. So really, like, is Lee et al. Buying us anything here? The lengths are much longer. So let's try and understand what's going on here. So it's a concept of leftover information that can tell us, like, give us an idea why Lee et al. Intervals can be so poorly behaved. And is it that Lee et al. Really isn't telling us much beyond sample splitting? Let's see. So let's look at a very simple example. Forget about regression. You have just a single Gaussian data from a Gaussian distribution which has mean parameterized by mu. Your selection is just a thresholding selection event. You report if y is greater than lambda. And Lee et al. Will look at the conditional law of y given y greater than lambda. Now to compare it with data splitting, let's just double the sample size. Let's have 2 observations and we report about mu if y1 is greater than lambda and y2 is greater than lambda. So let's plot the leftover information in the data from selection with the parameter values. And we see that Lee et al., you can see that sample splitting always has information equal to 1 because that's the variance. You would base your inference on the law of y2, unconditional law of y2, which is Gaussian. It has variance 1, so that is the leftover information that you have. Whereas in case of Lee et al., the leftover information can go to 0 when your parameter values become more and more negative. So as your parameter actual signals become more and more negative, that's when your selection becomes rarer and rarer, and after a point you're just left with no leftover information for inference. So we can see that Lee et al. Is certainly better than data splitting in this regime where you're actually using all the information that you had in your data and selection and you have no leftover information for inference. But it's doing better than splitting in this regime. So what is it like principally? How can we do better than both Lee and splitting? Yes?
I lost something. What's left?
So leftover information is actually when you write out the conditional law or the law on which, so in Lee et al. It's the conditional law of y condition on y greater than lambda. In case of data splitting, it's just the unconditional law of y2, right, which is just a Gaussian. So leftover information is basically the Fisher information that you compute based on that law. So once you have that law, you can take double derivatives and compute the Fisher information or the expected Fisher information.
And this is sort of the minimal variance of an unbiased estimator, this kind of thing, or something like. What I'm missing, I think, is why leftover information, having lots of leftover information, is a good thing.
I mean, intuitively, like what sample splitting was doing was discarding the data that you used in selection, but conditioning in some ways is trying to still use some information in that data that was left over from selection. So if you have, intuitively just think about it, the more the leftover information that you have from selection stage, you can use it in inference and get more powerful inference.
I think the intuition is fine. I think the problem is many of us don't know the
Formal
the expression or what it is.
Yeah, the expression is Fisher information. So the law that you base your inference upon, like I said, in case of Lee et al., it's a conditional law. You compute the Fisher information. That quantifies the leftover information that you have for inference.
Is the difference between some Fisher information that you had at the beginning and
I don't think not necessarily. Not necessarily.
Is it a major derailment if you just wrote it out for this simple experiment on the board?
No, of course.
It's not too much of a detour?
No, I mean, like I said, it's. So in this case, I mean, your law is, say, y minus. Right? So the log likelihood that I have in Lee et al. Is the log likelihood as a function of mu is this quantity, and my leftover information, this is the, I mean, it's actually, that's right.
Conditioning on is the minimum of the y's. sample splitting that's just conditioning on y1 being lambda, and non-sample splitting is conditioned on lambda.
So if you take the double derivative of this log likelihood, that's your observed Fisher information, right? That's what I'm plotting here. So all I'm saying is that observed Fisher information goes to 0 as your parameter value becomes more and more negative.
I think what I'm missing is, like, I understand when you use information sort of informally as an English word, like more information is good.
Okay.
I'm with you. Maybe.
You don't see where
What I'm asking is sort of like, why is this
Fisher information,
like, why is this the thing that we want, that we care about? Yeah. Why this specific measure of information?
But for-
I understand sort of there's some, there are various connections to sort of minimal variance, unbiased estimation, and things like that. That theory, like, I've seen it. How much of it I remember online isn't that much.
How
but
much
like,
is left over?
but Fisher information kind of, like as the name suggests, it's quantifying the information in the law that you're basing your inference upon, right? It's how informative is your inferential law about the parameter that you're inferring about. That's what Fisher information quantifies for statisticians. I don't know if I have a better answer for it.
There's a theory of asymptotics that says that literally the best estimators possible depend very precisely on the Fisher information.
unbiased.
I mean, in the classical regime,
No, just period. Any estimator, asymptotically, Fisher information governs its behavior.
If you say symptotically, unbiased.
No, just period.
Like
asymptotically.
Conditions, otherwise you get like.
Asymptotically normal Fisher information determines the covariance, like the Bernstein-von Mises type stuff, right?
Yes, local asymptotic normality. But it's just there's a theory that says in all classical problems, asymptotically the Fisher information described very precisely how well estimators
How,
could
like
possibly
the
behave.
minimal variance.
No. Estimator, period. Any estimator at all. Nothing about unbiasedness.
No, but just, okay, but
No, Adam is zooming in
no.
on the word how well, on the phrase.
like how well it
I
does.
think maybe an answer could be Cramer-Rao or lower bound also, like if you're, that's,
I have a feeling this is too much of a digression, I'm sorry.
no, I mean, I appreciate the questions, but,
Like, again, I get why, like, at some very
Sorry, dear God.
straightforward
I'm gonna write
level,
down a theorem.
information is good, and
Here's the theorem. For all losses, L,
yeah,
the limit, morally, infimum, as n goes to infinity,
yeah,
the best possible estimator that you could have,
yeah.
and then a supremum over all parameters theta that are within basically, so theta 0 is your true parameter, basically something like 1 on square root n of this true parameter, expectation under the distribution at theta of your loss of square root n times your estimator minus the true parameter. This is bigger than or equal to expectation loss of z, where z is normal 0, inverse Fisher information. That's the sense in which Fisher information
Sort of characterizes around in the neighborhood of a particular theta 0 what your estimation is. So
Yes.
now the expression in blue over here is some expectation of that.
Although
I mean,
I think
you might like, I mean, if you take the double derivative, that's free of y, so you don't even have to take the expectation based on this law, like here.
I think, Aaron, that your criticism is valid in, or I'm sorry, Adam, your criticism is valid in the sense that we're not necessarily doing asymptotic things here. So
It wasn't even a
with one
criticism, I
sample
just
and-
wanted to know. Like, I wasn't trying to be critical, I just wanted to understand, like, why is this plot on this?
Oh, okay, fine, all right.
Sorry.
I will criticize. I will make the criticism that the Fisher information has many beautiful asymptotic, or it's sort of the right asymptotic summary of how much information there is. However, usually we're just using one observation of a truncated Gaussian to construct a confidence interval, And I don't know of any real bound that you could get on the length of that confidence interval from like a Fisher information type thing.
Okay.
I guess maybe in meta-analysis, if we're accumulating many truncated estimates of this form, like a selection-adjusted meta-analysis, then I think you would have some interesting implications there. The way that we're typically using this in this field, I don't think we can give you useful.
But I think the main takeaway is, if you kind of understand Fisher information, is really just telling you how informative is your law for inference for the parameter that you're trying to infer about. How informative is that law? And it's sort of a quantification that we as statisticians use.
Why don't you go on?
So clearly we see that, like my main point here was that Lee et al. Basically somewhat uses all the information in your data that was in selection as your parameter value becomes more and more negative, as a result of which if you construct intervals corresponding to this regime, they become infinitely long because you don't have any power at that stage left for inference. Now, so the question is, splitting really doesn't look at all of the data in selection. It's winning in this regime because it's somewhat not looking at all of your data. It's just looking at a part of your data. That's why it does better than Lee et al. But there is this fundamental problem in splitting that it's discarding that data completely, and hence in this regime it's Lee et al., which is, in the other regime it's Lee et al., which is winning because it's somehow leveraging on the fact that it's still using some information left over in the data from selection. So how can we get the best of both the worlds, both splitting and Lee et al.? And a simple answer is carving. So why not we combine the idea of splitting and Lee et al., that is, we use not all of the data during selection, but just Y1 for selection. But now we just don't discard it completely, but still try to use some leftover information from it in inference. So we try to look at the law of Y1 and Y2 conditional on Y1 greater than lambda. So we are using a fraction of the data for selection, not looking at all of the data in the selection state, but still we are using the conditioning idea of Lee et al. To try and utilize whatever information is left over to get the advantages of conditioning. So this is a plot of, again, the leftover information or the Fisher information of the conditional law. So this is what I've added, which is the Fisher information from the carved law. And you can see that it dominates both splitting and Lee et al. Mostly. So it's probably the right thing to do, which gives us a best of both worlds, both splitting and Lee et al. And as I view it, it's fundamentally the right approach to go about. So if you want to have inference with some reasonable power as your end goal, don't look at all of your data, reserve some of your data for inference. But at the same stage, we can condition on the part of the data that we use for selection to give optimal inference. So the takeaways from this part of the tutorial that I hope you took is, I don't want to view data mining really as a bad thing to do. It's not data snooping. There are like efforts of large communities to explore data and get some structure in data. So we want to leverage upon those findings and do a confirmatory analysis to somewhat give confidence intervals and reasonable point estimates with some statistical guarantees at the end of the day. Secondly, I talked about this conditional approach to overcome bias. Hopefully, I conveyed you the guarantees of conditional approach, and I gave you some first approaches to overcome selective bias, which was data splitting and Lee et al. And we saw, maybe I didn't convince all of you, but maybe if you get at least an intuitive understanding of why Lee et al. Can lead to infinitely long intervals, because it doesn't have the conditional law once we use all of the data in selection. In some regimes, as selection becomes rarer and rarer, it doesn't have any more leftover information for inference. And in the second part of the tutorial, though I already gave you the answer that carving is the right way to go, I'll talk about it more in details and what can be the technical challenges in providing carved inference and how do we overcome them at the moment. So that would be in part 2 of the tutorial.
Good, thanks. I propose that we resume at 10:30 as scheduled. Shorter break.
Subject to Snigdha getting enough of a break. Yeah.
Oh no, I'm good.
She's the best.
Uh-huh.
Yeah, well, that's fine.